Hello, everybody, and thank you for coming to this session. My name is Tania Pereira Chilima, and I'm the Chief Technology Officer at Univercells Technologies. And today I'll talk to you a little bit about our dual platform for virus manufacture for both adherent and suspension cell-based approaches. So I'm part of Univercells Technologies. We are a subsidiary of Univercells Group, funded in 2020 with the mission of revolutionizing biomanufacturing by delivering best-in-class technologies. This is just a little bit of an introductory slide. So I think, as we all know, the gene therapy market, it's highly valued at this. It is continuously growing, and one of the key characteristics of this market is that the annual demands for different indications vary significantly according to the dose size as well as the incidence rate. And some of these indications require very, very high annual demands, which means a lot of viral vector to be generated. So we need, in this sense, both flexibility to accommodate different annual demands while still being able to tackle those very, very high annual demand indications. And this slide just summarizes the different needs of the gene therapy market when it comes to technology, some of which I've already indicated, but going more into it. So capacity and high performance. So as I mentioned, the ability to cater for those very high annual demand indications is important in the technology selection. And capacity, we're talking about physical capacity of the reactor, but and when we talk about performance, is actually a system that enables you to increase your productivity. Flexibility, as I mentioned, there are different annual demands for different indications in the space, but flexibility also means the ability to accommodate different process designs and potentially expression systems. Automation, when we go from the traditional technologies such as flasks, we see that the need for automation is very important in order to have a scalable and robust process. Ease of adoption is also important, as we see a lot of different products being developed in more manual technologies. So when we move to more scalable technologies, that transition is very important. Following that is the scalability. So being able to reproduce the results attained at the small scale, at the large scale, greatly benefit also manufacturers. And finally, low capex and low footprint. So when we look today at the technology landscape for gene therapy products, we see that there's two types of technology. So you have the suspension technologies, which are more stir tanks, and then adherent technologies. When we look at suspension technologies, they have both pros and cons. Of course, they're well established, they are multi-source, and they can go up to very high capacity. But at the same time, with technologies, the volume creation pretty much increases linearly with the capacity of the technology, and also requires rather a lot of know-how to first adopt and scale up the systems. Looking at the adherent landscape, we have both intensified packed bed bioreactors and flasks. Of course, flasks are routinely used in labs, so it's quite easy to adopt, but the lack of automation and scalability is a challenge. But when we look at packed bed bioreactors, indeed, they are able to increase the capacity, but they haven't quite been able to cater for very, very large annual demand indications. So then when we look at this landscape, what we thought to do was to deliver a technology that can both reduce the operational volume while still having a high capacity. And how did we achieve this? We achieved this by applying the principles of intensification and chaining. So what we are doing is that we are moving from standalone unit operations in, combined together using intermediate tanks to the intensification of processes through biomass immobilization, and the biomass involved adherent and suspension cells within a structure fixed bed bioreactor, which is what you see there. That structure fixed bed bioreactor still provides the homogeneous environment that you would see in a suspension cell culture, while enabling intensification of the manufacturing process. The other principle that I just mentioned was chaining, and that means linking different unit operations together. By doing so, you're reducing the size or number or actually removing it all, the intermediate tanks across the different or after the different unit operations, while automating the whole process and reducing the overall footprint and capex. So this is our technology portfolio, which basically illustrates in practice these two principles of chaining and intensification, starting with the ScaleX bioreactor, which is a structure fixed bed bioreactor, available in multiple configurations, going from the proof of concept scale, which is a hydro bioreactor that you see there in the left-hand side. Then in the middle, we have an intermediate scale, which is quite a unique configuration for a packed bed, for a fixed bed bioreactor, which is a carbo bioreactor, available in both 10 and 30 meters squared configuration. And this bioreactor it enables both to make the bridge between the proof of concept and the larger scale, but at the same time is also available in a GMP format, which means that companies don't have to oversize a manufacturer during clinical trials. And for some indication, this might just be the ultimate scale that they'll go to. And finally, we have the nitro bioreactor, which is a really large-scale commercial bioreactor with 200 to 600 meters squared. The nitro bioreactor is incorporated into the Nivelles line, which is where really the chaining principle kicks in. Where you have not only the bioreactor, but additional unit operations such as clarification, concentration, diafiltration, etc. And this is all managed by a single automation system with a single HMI. So just drilling a little bit down on the bioreactor. So key features of the system, if you have this kind of cartoonish view of the system, is highly intensified. The highest system has around 10 meters squared for cell growth per liter, scalable and reproducible homogeneous environment. So we have the same structure fixed bed design across all the bioreactors, low shear, and it has all of the controls integrated into the bioreactor. So during the process, we basically decouple the environment where the cells are from the large volume of media needed to feed the cells. So what we do is that during the process, we either recirculate the media in and out of the bioreactor or perfuse the media in and out of the bioreactor. And this is very important because while the cells are staying in the bioreactor, we are able now to control the amount of media and the volume that we want to process downstream processing wise by decoupling the volume of media from the cell containing environment. This is also important in terms of flexibility because all of a sudden we are able to perform recirculation or perfusion processes without the need of an ATF system. Within the bioreactor, the media circulates upwards through the fixed bed and then falls back again into the falling film. Within that path, what happens is that we design a unique dual layer structure, which has a polypropylene mesh, which is what you see here in the middle, in the right-hand side, combined or concentrically rolled with PET fibers. The polypropylene mesh has very large porosity, which provide these preferential pathways for media flow, while the PET fibers is where the cells are growing. Now, by doing so, what we are doing is that we are decoupling the direction of the media from the cells, which means that we are reducing the overall shear that the cells are experiencing. By doing this in a structure fixed bed design, what we are doing is that we are enabling a reproducible low shear environment across batches and across scales, which is why we see some of the results that we have in the ScaleX bioreactor. To illustrate the structure fixed bed layer, or sorry, the structure fixed bed design that I just explained, this slide here just shows how the polypropylene mesh and the PET fibers are rolled together. And what you see there, and they are rolled in a 2-to-1 ratio. So you have 2 PET fibers, 1 polypropylene spacer layer. And what you see there is how the cells are growing at the bottom. It's a very small picture. How the cells are growing along the fibers in the PET fibers. You also see the cell density distribution in points taken at different height and diameter points within the PET fibers. And you see that, you know, there's quite a rather narrow range in cell densities. So on scalability, the ScaleX system is scalable by design. We designed it to thinking about the environment the cells are experiencing and to maintain those across scales. So keeping the same gas exchange dynamics, so KLA and CO2 stripping, the same linear speeds for nutrient delivery, the same cell to volume, the same volume to surface ratio. This is very important, especially in batch steps like a transfection, ensuring that the overall cell concentration in the bioreactor is the same across scales, and homogeneity. And I already explained that to you that will be through the fixed bed compaction, as well as through the height of the fixed beds to limit formation of gradients. So when we designed the bioreactor, we basically kept 2 different diameters and 2 different heights. And that's what you see there. The ScaleX carbols have the same diameter. The ScaleX nitros have the same diameter. But going through from the carbo 10, which is the smallest carbo, to the nitro 200, which is the smallest nitro, you basically scale in diameter, so increase the diameter, and the same thing when you go from carbo 30 to nitro 600. Now, we actually run cell cultures with all of these with bioreactors from the 3 families, let's say, the hydro, the smallest one, the carbo, the intermediate bioreactor, and the nitro, which is the largest bioreactor. And this is what you see there. This, for example, this example is of a viral cell culture, for 5 days. And what you see is that the growth is rather reproducible across the scales, going from the smallest family of bioreactors to the largest. And today, now I'm just going to show you how the system was applied in a couple of projects for both, or 2 case studies for both adherent cells and suspension cells. The first one, it's an adherent project using HEC-293 cells for a transient transfection process for AAV production, and here we demonstrated high productivity, which was 4-fold higher than traditional packed bed bioreactors. We have demonstrated reductions in materials costs, so we were able to basically reduce the transfection density by 3-fold, and by that we reduced the amount of DNA NPI by 3-fold. And finally, we were able to reduce the seeding density while still achieving very positive results. And with that, we can reduce the inoculation density and basically intensify the seed train as well. Looking at the case study number 2, and this is where we use suspension cells for adenovirus production. Here, the key take-home messages is that we achieved the target productivity, and we actually surpassed the target productivity. We achieved the target cell entrapment and suppressed that as well. And finally, we achieved the same or better population doubling time than the control. So on the results of the first bioreactor, of the first case study, we basically used 4 bioreactors, and with each one of the 4 bioreactors, we made changes to the process, ranging from seeding density, transfection density, lysis region, and also the volume of media needed for the cell growth. And what we saw is that the cell growth profile was the same as the one in the control T flask. And when we look at the productivity, so the red bars there, so the range in red are the productivities achieved with the ScaleX bioreactor in each of the 4 experiments that were done, whilst the light gray is the reference process in cell factories, and the darkest gray is the traditional packed bed bioreactor process. And here, what we see is that we achieved comparable results with respect to the cell factories, even though we reduced the amount of cells in the system per square centimeter and reduced the seeding density and media usage, while as we achieve a 4-fold higher productivity with respect to traditional packed bed bioreactors. When looking at the second case study, and this one is using suspension cells for the manufacture of adenovirus, this was an infection process. There, what we see is that we did 3 bioreactor runs. First one was just copy paste of the traditional process. The subsequent runs were both adapting the seeding density and prolonging the growth phase, or sorry, the production phase. And on those results, what we saw is that the per population doubling time was reduced when moving to the ScaleX bioreactor versus the shake flask, and it was more robust. So the variation was a lot more narrow. We also see that actually, although our target was to have 80% entrapment, we had 95% entrapment of the cells in the fixed bed. On the results, we had consistently achieved a higher productivity, specific cell productivity with respect to shake flasks. Just to complete the picture, I've presented on the AAV process, I presented on adenovirus process, but we also have generated results using lentivirus processes, where we again saw a higher productivity than traditional packed bed bioreactors. And finally, just to conclude and to close the loop on the presentation, with our system, we believe that we can provide that ideal system that I was referring to you at the start of the presentation, which combines both low footprints and low volumes with high capacity through performance and actual physical capacity, while still having all of the other benefits that are needed in gene therapy manufacture, such as flexibility, scalability, automation, etc. For product developers, this means that there is no rush to select and transfer the platform at very early stages of the development process, because we have demonstrated the ease of transfer of the system from flasks, both shake flasks or T flasks, to our system. And for CDMOs, this also means flexibility to accommodate suspension and adherent cells in the same system, which could mean higher utilization of a room, because you can use the same system for more projects, basically. Thank you very much, and I welcome any questions at this point.